Good morning and good evening to everybody, depending where you are. So, as Bijoy introduced me very nicely, I tried to uh, um, uh, guide us as the brain is working, as we know at this time. And I do it is as a simple-minded, and this will be a simple-minded way. And first, I'd like to start with this Ken Hill citation because I think it is pretty valid these days. Uh, if the brain were simple enough for us to understand, we would be too simple to understand it. So, but we are pretty keen to know what our brain, the central part of our life, is working. So, because as Bijoy told that I'm an anatomist, and therefore my background is depending on the aspect that the structure and the function goes together in hand. So I'd like to first show what I know about the brain structure. And here I show you that so-called rainbow brain, which was genetically altered, and they are shown up different type of neuronal cells, pyramidal cells, granular cells, and the location distributed along the cerebral cortex. And in this image also we see that is the distribution of the neuronal cells isn't homogeneously scattered inside the cerebral cortex. There are some accumulated area where the numerous cells are closer. And the other part of the brain is the cerebellum that shows the by immunohistochemical staining the big Purkinje cells and the granular cells and the neuropil, where the connection happening between the Purkinje cell dendrite and the granular cells and the incoming afferent cells as well. So in the long, as everybody has a long history, so the brain research and the brain histology also has a long history. Here is the Cajal, as he used silver impregnation to show cells on the in the brain. So there's the Purkinje cells. You can compare how it looks like differently. And here also Cajal images one this all silver impregnated images, the pyramidal cells, and the synaptic button on neuron, where the afferent information arrive and communicate with the neuron. And here is the dendrite, the dendritic spikes, which located around here in the cerebellum and in the cerebrum, the upper cortex, upper layers, but everywhere, all layers, all 6 layers, there are dendritic spikes and accept afferent information. And the new technique, the electron microscope, where we can see synapse structures and the other dendrite images in the very close to each other. So there is a very tiny extracellular space that has among the different structure, the brain. So there are two big different structure that the neuronal cells can communicate each other. One is the chemical synapses, shown here, the presynaptic and the postsynaptic structures, and full of the synaptic vesicle. That vesicle contains the different neurotransmitters. So the glutamate, if the is stimulative, and the GABA, there is the inhibitory synapses. And the other type of communication each other, the gap junction, which is very different from the chemical synapses. It's very tight. The two cells cytomembrane, and there is the electrical communication is going through in this gap junction, as we know at this time. And we, in this part, we can see the connection between the neurons, astrocytes, and the oligodendrocytes. And the interesting thing is that in the gap junction, the neurons has a specific protein, that's the connexin 36. The astrocyte has a very different connexin, so a very different proteins, connexin 43, 26, 30, and the oligodendrocyte also has a different protein, connexin 29. So it means to me that the protein has some very specific function in the biological system. So the neurons and the astrocyte and oligodendrocyte, which define probably the functional connection difference between the neurons through the protein, through the different proteins. So here, again, a kind of historical image when the old people, the neuronal pathways sketch through this line drawing, showing the different part of the brain, and then is line shows the connection between the different structures. A new technique, this the diffusion tensor image, shows how the axons, the neuronal fibers, show and can be seen in the brain. And then we can imagine that at the end of every strain, file has a neuron, a cell. So because that is a connection that's made a neuron through the axons, which can be connected with the two hemispheres and in the intracortical connection. So it's very different orientation. This hardly can be seen. We can say that everything is connected with everything. It is just our intention, and we are trying to distinguish which brain area has been connected with the other brain area. A network connectums design is a new standard or new technique where the structure only named and not shown the brain structure, but only with the name. And there is also line connection between the different area of the brain. And this shows that there are some center where the connection is more diffuse, and this is such called a hub type of center, which start the central important center because the information coming into that space. Of course, the people, as they work with the brain, the histologist people, they had a different histological technique. Golgi impregnation, Nissl staining, simple, and the myelin staining. And they showed that somehow there is a kind of vertical orientation inside the brain, instead of seeing in particular the simple stain, the Nissl or hematoxylin stain, the cerebral cortex shows a kind of laminar layer type of structure. So, and this was layered 6 layer in the cerebral cortex. But when the myelin staining shows this vertical image, of course, was the thought that, oh, well, so many neurons and the myelin, which connect these neurons each other. So how the brain is functioning. And Vogt, a German histologist, thought that there will be a vertical oriented connection between the neuronal cells, and instead of the horizontal layering. But in this picture, we saw that the vertical and the horizontal area, there are very many fibers. So the connection is just incredible dense in the cerebral cortex. So going on this way, Szentagotay, a Hungarian anatomist, also try, oh Jesus, uh, uh, notice this vertical orientation, and he uh, proposed a kind of functional module which is vertically oriented, and there is two parts of this module. One part is here, which accept the afferent information, it's incoming information, and the right side is the efferent information, which has been cooked inside this functional module, and the efferent information or efferent order going out to the other functional columns. So, and of course, how this functional module is working, depending on the uh, kind of neurons which have been built this functional module. If this more excitatory component or less inhibitory, then the outcome is different. And so the outcoming information, depending on the building bricks, how they can function together and digest the incoming information. So he also make a sketch that he thought that this functional module, which has about the 200-300 micrometer column diameter, has a very versatile connection with the other functional column in the same hemisphere, and even it goes to the other hemisphere. So it's as connected as the so-called mirror modules in the brain. Is can be seen that intercortical, gyral connection with the functional module. So if I turn now a little bit to the functional aspects, how the brain functioning, I think that it is worth cite this Bertalanffy sentence. It is necessary to study not only parts and processes in isolation, but also to solve the decisive problems found in organization in order unifying them, resulting from dynamic interaction of parts and making the behavior of the parts different when studied in isolation or within the whole. I think the problem we can see that it is pretty hard to isolate this functional unit and to check what the different functional module scattered or can be found inside the cerebral cortex, what is the characteristic function of a single module. It is pretty hard, and therefore we just so-called the whole activation or activity of the brain can we see, and the electrical activity of the brain, then we can detect that electroencephalograph that was discovered in 1930 on Berger, and then he noticed a different kind of electrical wave. That's the beta, the alpha, theta, and the delta wave, which can be seen very differently according that the frequency is very different. If we show the electrical waves in logarithmic scale, and not the 10, but the natural logarithmic base, which is 2.16, then we can see that the different brain waves overlap each other, which has probably the purpose that the different brain wave which produce the different neurons can spread to the neighboring cell under some circumstances, because this is not closed. These brain waves is kind of open wave that goes into the other type of wave very easily. That maybe could be the base of the so-called open system, which has a very fluid transition from one cells to the other, and then probably the neuronal network much more easily can be established. So and then the oscillation among the neuronal cells can be built up easily. So, but the oscillation has needs a timing, because there is a wave needs of the wave run through the timing, and the synchronization can be established when a good timing is happening. So the interference is very important. This I like this Saint Augustine citation, which I borrowed from Bujaki. So if the past is over and the future has not yet come, all that exists now. So how long does no last? So it's very interesting in our days, but of course it important in the brain wave, how the so-called overlapping brain waves can go each other. So it turned out during the very intense research that the brain has some time conductor, so which is the uh, suprachiasmatic nucleus has this function to so-called synchronize the function, the neuronal function, electrical function in the entire brain. So the suprachiasmatic nucleus uh, organize a kind of circadian rhythm, which is around 24 hours. And this has been uh, made about 20,000 neurons, which has the suprachiasmatic nucleus. And again, we noticed it was a kind of maybe not surprise, because we know that the protein and the lipid build up the biological structures. So during the research, uh, very specific protein, the pair protein, was isolated from the suprachiasmatic nucleus, which uh, supposedly make the circadian rhythm uh, depending on the protein electrical and electrostatic properties. In the rat also discovered this protein, this the CLK, the circadian locomotor output. So so how important the group activity shows that when they isolated the suprachiasmatic neurons, nucleus neurons, and they put in petri dish, they didn't see the circadian rhythm. It was 20 and 28 hours rhythmic activity in the tissue dish. So in the brain, in situ brain, the nucleus in the group neurons somehow synchronized everybody's electrical activity about the 24 hours, maybe plus minus one, but much less than the 28 hours in the singular neuron. So that the group activity is looks pretty different from the individual neuron. We notice this in our society as well. So if we I just like to bring up this side because the neuronal ELAV protein was discovered that this this protein family, which has a different type of a different family in the human, the ABCD human protein families. And this protein probably has the very, very different functional neuronal pathways. So the visual pathway has a very specific ELAV proteins. The auditory pathway has a different, and the olfactory pathway has also a very different, which has uh, function to recognize each other during the migration, for example. If I turn now to our interest in the auditory pathway that was also discussed in the previous seminars by Bharani and Prem. And here we can see, so the peripheral part is from the hair cells until the auditory nerve enters the medulla, and then the central part, which is the receiver of the sender information, the sender part of the hair cells, and the conductive area, this different neuronal chain, which leads the information to the primary auditory cortex. And to study the primary auditory cortex is discovered also very distinctive location, the different sensitive neurons which are receive the different hertz sound electronic package information. And then here we see the so-called speech network, which the circle is probably, we can say that the primary auditory cortex, the secondary auditory cortex, the Wernicke area, and called sensory cortex, and the efferent size is the Broca area, the motor auditory cortex, which is connected with the motor and the sensory cortex as well. Here I just brought up the telephone that our telephone was discovered and developed step by step. Alexander Graham Bell, and 1877, and here we see the three important component, the sender component here, which actually transfer the sound wave that the person through the speech relieves, and this sound wave enter this box where electromagnetic circuit has been, and according to that, how the frequency is enter this box, the the wire conduct these electrical signals to the receiver, and the receiver transform back the electrical signals to the sound wave, and the person can hear the sound. And if we compare, and here is Graham, the first design. So the sender, the wire connection, which can be iron or copper, because the copper has a different property and conduct better the electricity and wire. So, and noise signal connection is better. So developed, and here is the used telephone. But in the so-called biological telephone, which can be take as the hair cells, the senders, which receive the auditory waves, this energy waves, that presses the hair cells and transfer somehow these sound waves, electrical signal, and this electrical signal push through the neuron chain up to the primary auditory cortex, where is the receiver. And we've heard Bharani lecture that in the hair cells also very specifically receive the different hertz of the sound by means of having different protein structures inside. And if this is the case, then we can probably take that the primary auditory cortex, the different neurons which are sensitive, also receiving the different sound frequency, has different proteins, because that allows them to receive and and resonate together with the electrical frequency of the sound was incoming to it. So it's very different from the mechanical telephone, the biological telephone. So we definitely have to work with the different protein and research what this protein electricity has different in the neuronal activity. So to go how the primary auditory cortex is working, that was Prem from the found this paper, this Swan neurosurgeon, who was working with epileptic patients. And today the epilepsy treatment is to figure out where there are the focus where these epileptic bursts starting inside the brain, and therefore uh, not the electroencephalograph is using, but open the skull, and the electrode has been put in the auditory cortex, so this called corticoelectrograph, and they receive the activation which is going inside the cerebral cortex. So the neurosurgeon uh, who has been interesting in the sound and the speech construction, use this patient and did the experiment that they ask the patient to pronounce the different phonemes, uh, and then they recorded the underlying structure, neuronal structure activity, and they compared five different electrodes, and they showed the activation intervallum or duration at the different electrodes. And if we see the timing, which is 0.2 milliseconds, and then we can see that every electrode has at this time point very strong activation. And if we compare this horizontally, then we see that somehow the first and the second electrode at this point, so synchronously active. So it means that under this electrode, the neurons uh, build up a cluster and oscillate together because the timing is the very same. So simultaneously they are active. And then we can see here the 3, 4 electrode under the electrode, the neurons activated. So this cluster have been built up to handle the incoming auditory information. Of course, what is this handling and what happening and inside the neuronal clusters that we don't know exactly. We just can detect that those cells together as cluster active. So of course, because the animal experiment is much easier to carry out, so it's easier to do experimental as animal. So this group has a cat that was the electrode, 2 pairs of electrode put over the auditory field. And in this case, not only just put on the surface, but the electrode was deeply pushed inside the cortex, and they constructed these electrical grids that they if they push down to the cerebral cortex, probably the electrode reach the second and the fourth layer of the cerebral cortex. And here also they make some noise to the cat, and they recorded what's happening. And they also discovered that there are some electrodes that synchronizing the activity, and they found 4 different clusters during the different sound stimuli. And they showed the diagram here and the spike activity about again showing the same timing. So that was the principle of calling as this clustering activity. And they noticed that if they applied different sound, then the clustering size moving smaller or larger. So a very dynamic clustering happening depending on the stimuli. Where the stimuli is complex, the clustering size will be larger. So it means that more neuronal cells asking the neighboring cells to help us to handle the incoming information. So so far the different histological technique, the anterograde, retrograde tracing, the researcher discovered a very diffuse connection between the different brain areas. And here are the parietal cortex has the very diffuse connection, the temporal, frontal area, and the occipital area. And here shown the prefrontal cortex also has a mutual information with the different areas. I just put here the visual cortex, which is very interesting. So not only the primary visual cortex, which process some visual information, but there are some supplemental additional helping visual cortical information, the secondary, tertiary, quaternary visual areas, which are which are processing the different information. They got it. And there are 2 longer pathway, the so-called dorsal visual pathway, which coming from the occipital cortex and goes through the parietal and landing in the prefrontal cortex, and the ventral visual pathway goes through the temporal cortex and also entering to the prefrontal cortex, that we think that the thinking and decision making happening here. So, and of course, when we communicate, not only the auditory information enters in our brain through the ear, but we see the person, so visual information also enters into our brain, and that the brain as a open system, as Bijoy has also mentioned, but not only just an open system, but a self-organizing system, which which has a very internal route, how the brain handles and working out the visual and the auditory information. It has been combined that we can put parallel and say that this coming from the same person. So the new technique developed, the fMRI, the functional magnetic imaging, which shows us the surface activation of the brain, the cortical activation. And here are these 4 different imaging, which shows a very huge, extensive, different area of the cerebral cortex, which is active when we hear words. So not only just the primary auditory cortex active, but this is the parietal, occipital, temporal cortex border, and here is about the sensory and motor cortex activation. So is a huge number of cells got activated synchronously. So in this image, seeing the word, different cortical area active, mostly the occipital, the visual cortex are active, and very, very faint or no activation in the primary auditory cortex. So it's very, very interestingly a different area has been synchronously active. The speaking words also has a different image. Here we'll see the sensory motor area and the Broca area, but also as the Wernicke and the temporo-occipital area is active. So, and very different area are active when we generating words. So creating words or and the new words has been established, is the frontal cortex is the most active in this area. So this shows us how the brain as a whole, as a self-organizing structure, has been synchronously active, and as it is dependent, independent from us, we don't know how this happening. This is really a self-organized system which work through the pre-wired different neuronal pathways. So,
Bella, I'll just interrupt you saying that you have 30 more slides. So see that each slide you take a minute or so that we can.
yeah, no, that so
Yeah.
this I show that the, as I told before, showed before, that the coupling, the hostile and the information between the neurons going to chemical synapses and gap junction. And here is the electrical recording from the electrical synapse and the chemical synapses going on. It shows that. So here I just brought back to the Santa Gotai proposed functional modules. It nicely can be imagined that somehow in this different huge area active through this accumulated functional module coherently synchronize each other. So to uh, uh, note notice that we used to say the neural cluster and neural assembly. And so in this, what I found in this paper, it was told that the neural cluster inside the cortical area boundary, so it respects a very small cortical area, and then spatially very closely related each other, and then is dependent on the stimuli condition. A neuronal assembly, which is said that temporally working together, correlated unit activity, and different large area can be activated, and we called that the neuronal assembly. Uh, the clustering uh, has two types of clustering. That's spatial clustering, then distributed in a neuron density, as I mentioned, that in the cortex there is no homogeneous distribution of the neuron. There are some accumulated area. So but the subcortical area and the thalamus, there are very distinct neuronal cell accumulation. And now, as we actually don't know very exactly how this clustering happening, and new intense research is going what neurons characteristic may play a role in the clustering. So they research and the neuronal morphology, the connectivity, the spiking, the functional dynamics, and neurochemical content, gene expression, and the gene expression. Beside that, the so-called transcriptomes, sync cell analysis is happening, which is the transcriptomes is depending on the RNA research and RNA content, which determines the protein content of the neurons. And here I'm showing that there was a group that put the question whether if they put very primitive progenitor neurons in the cell dish, whether the neurons could develop. So it means that they really inherited messages having to develop the different dendrites and axons. And here also different, the red lateral geniculate nucleus described, and they found a very morphologically different projection neurons. And here interneurons, different morphology shows in the dorsal prefrontal cortex in the monkey. And here is some electrophysiology of these cells that really they somehow morphologically, which probably it means that the building protein determine the morphology, but the building protein define the electrophysiological activity and can be a fast spiking neurons, medium fast neurons, and slow fasting neurons. So this could be taken into consideration that they have a very different functions. Here also another group research where is the different morphologically different neurons clustering and projecting in the different brain areas. Here also see the 8 different clusters in the cortex, and then these different clusters have have very different morphologically appeared neurons, and these neurons projecting from the cortex to the medulla, and then has very different morphologically appearing neurons, the non-medullary projection, spinal cord projection happening. So it's a very interesting connection is between the functional, morphological, and clustering activity appearance in the neurons. So in the cochlear, ventral cochlear nucleus, that is belongs to the auditory system, also shows that there are so differently built up, morphologically different cell types are, and very different functional entities they have. The bushes have 600 microseconds frequency can be handled information, incoming information. The octopus cells more faster, 200 millisecond precision can be handled incoming information. So it's very, very unbelievable. So closing my presentation, I conclude with Joy here, as she said. So as with any tool in science, fMRI, complementary imaging techniques such as PET, EEG, optical imaging subjects are subject to limitations. Some of the hardest questions: how do neurons transform energy packets captured from the environment into conscious experience, still remain far out of reach. Thank you very much. That would be. Thank you for your attention.
Thank you.
So can you all unmute and a round of applause for Bela? And just unmute.
Thank you. Thank you.
Where is it? Unmuting. People unmute and okay. All right. One more time.
So
I'm not hearing.
So this is my simple-minded way to see the brain morphology and functional connections.
Right.
Can I ask a quick question to Bela?
Yes.
Last slide is beautiful. So, and also that relates to Bijoy. So, how do you define this conscious experience? Finally, we want to define an objective day. So all input, function, structure you define beautifully. It's a very complex system.
Yes.
Finally, are we going to define difference between an octopus and a human, or octopus also has conscious experience?
Yes.
Right.
I think that somehow we have to accept that if we look to the whole animal world, that every animal, lower or higher developed animal, has a very accordingly complex neuronal system that handles and support their life as a protective system, and has the surplus function that allows some thinking function, which we easily say that the instinct function of a bird, for example, that can fly or build a nest, but probably the instinction can work through the primitive cerebral cortex that the bird has. So, and the monkey brain has a more complex brain, has more neuronal cells, so and morphologically maybe very different protein build up the neuron, so the function is very different. And our brain also has this double function, that our brain protects us to survive in our surroundings, but we have some other mission to create something very nice, productive, thinking, as we just look around, and and this cerebral, our cerebral cortical function is that we discovered that we can build a house, we can build a train, we can build a car, and some others.
Yeah, right. Let me add something. It's a very good question, and it's relevant to what we're trying to do. The model is the following. The model is we have an operational brain that we are using, which is rather gross. The brain knows much more than what we think we know. The subtleties that you, Bela, showed, structures, details, morphology, cell structures, cell variations, they're all there in the brain to have an exact picture. As you are seeing me, an exact picture is painted inside of you. But that exact picture is not known to you. You are not using it. You are using a very gross picture, an operational picture, an application picture, an engineering picture, which is maybe 10% of the information that you receive. Now, why is that so, and how we operate with this information is because we don't need the details of the information, because that society, I mean, that definition I will not get into. So the question is, can we then understand what we know? That is, if the stimulus is Bijoy is speaking, you don't need to understand Bijoy as the person. So Bijoy's face is enough for you. But the brain knows Bijoy as the person because brain is deciphering also Bijoy's voice, Bijoy's words, Bijoy's emotion, Bijoy's accent, Bijoy's way of expression. And for you, they are not saying what Bijoy is saying. You are satisfied with, oh, he said this following. So that's the dichotomy. And so that we have to kind of work on, saying that there is something called cerebral knowledge inside the brain and something operational knowledge that we are using. So that operational knowledge eventually becomes our speech. Like to what we understood, not what we know about. What if we think what we know about, then we have to meditate and the brain itself may reveal. So that we think has happened in the past, that you can see the cosmos, skies, trees, rains, and then you don't know what is happening. You meditate on them, and then the brain will speak you to reveal you back that this is what we saw. Now we are not doing that. We can do an experiment where such things can be done, but what we are after is the whatever grossly we would know, we call it a tree. When we call it a tree, we don't go into details about what tree is, but the brain knows what tree is. So that's the difference. Something called exact knowledge, something called applicable knowledge. So we shall see. So the logic from the clustering, what you have to take home is, out of all this, there is the information threshold where our processing stops and we say we're happy. I saw my mom. I don't know my mom fully, but I recognize her. I saw my friend. So that kind of a operational handshake we're operating with. Now, Indian literature is big on this. What is true knowledge and what is local knowledge? And we'll go into that once we know the words, what a word is, how the speech comes, how much speech is truthful, what is speech expressing. So that will come. We'll gradually go into that. On this, hopefully, we will be doing a second seminar about how the stimulus forms into a cluster. We are still working on it. But anyway, other questions can come. I'm sorry that I wanted to say this, because this particular density of information that you have, what the brain has, so that Bharani told in the first lecture about the role of the brain. The role of the brain is to understand, to protect us. But protects us doesn't mean to communicate to us.
Not enough. No.
Protecting us because at given time, it will give us information to help us. But when we are secure, the brain doesn't care about. It says, okay, move on. All right. Other question. Yeah.
I have a question that to Dr. Kosaras, that functionally, what is the difference between generating a word and speaking? That two different parts of the brain are activated.
Yes.
What is the difference between experiment when we are performing experiments? What is the difference between speaking a word and generating a word? Because I saw that two different parts of the brain are lit up.
Yeah,
Yes.
Is here.
Yes, that's right. So in the last two, the bottom here, speaking word and generating.
Generating words and speaking words. Yes. So the generating words is going through the so-called in the mind function. We don't know very exactly what the mind is. Our experience so far that the mind, the decision, and the creative creation have been happening in the prefrontal frontal cortex. So it doesn't matter, and I don't want to express the word that I create. I just want to create a new word. So my dictionary will be increased. And maybe the next time I will use the word when I speak. And that's a very different neuronal area, which send out organized information to the vocal system, including the lung, which has the air thrust through the vocal cord, and in the mouth as a resonance chamber, and the tongue position define the different sounds to express. So it should be a different neuronal structure be active to send out this information. Instead, the generating word is just a internal remaining word in the brain, in the memory, so-called.
Thank you.
Right. I think that's a very interesting question, Jaydev. This actually goes to the heart of this whole experiment here. That is when we think, when we contemplate, I contemplate of you. So that process, all information about Jaydev in me are being probed. In Vedic literature, Indian literature, it has been called para. That is, information is there, we are probing them, we're bringing them in. We don't know where are they. So what Bela is saying, the neuroanatomical regions where is activated for a particular kind of generation of words. So it not necessarily mean that that's the only locations, but what it may mean is that the generation process.
So may I ask a follow-up question, Bijoy?
Wait.
Yes.
We just
exactly on
it.
that. So
Yes.
let's say when somebody recites a mantra, and when somebody recites in his head without speaking, then the two different parts of the brain will lit up.
Yeah, let me just complete this. Okay. So what happens then is that this particular thing, when we generate, so that generation, after being generated, question comes up, how do we communicate this? The communication comes through our phonetic system that we may have, which is limited. And then we can simply mutter them.
Yes. So
that we think can happen where some kind of a expression is created, which are translation of the inner image to acoustic image. Or what we can do is we can capture that muttering and then see if we have a local language and we can convert that thought into local language. So these things we will come later. And your next question is whether two regions and so on, let Bela handle it.
Yes.
Yeah, can I before the answer comes, can I
respond
Yeah,
to your second question? I think that somehow, so I noticed by myself when I read a book and read the words and the sentence, I somehow feel that my vocal system is kind of steady state condition, as if ready to make action, but it doesn't know that it doesn't do that, but ready. So I feel that when I read this sentence, somehow I feel in the different muscles that maybe some afferent information is there, but somehow the last moment, the action is somehow blocked.
Thank you.
I also have a little addition in answer to Jaydevji's question. When you specifically say mantram, it is not something new unless you're reading it for the very first time. But generally, when you say mantram, you have heard it before. So there is a memory of the sound. So when you are reciting a mantram inside your head, you also are hearing it inside your head. And that's not what is captured in these fMRI pictures.
I see.
So the generating words in this case is not recalling auditory sounds. This is generating a word based on showing a picture. So you show them a picture of an apple and they are generating the word apple.
I see. Okay. Thanks, Bharani.
Can I share something?
Yeah,
on
please
this
do.
point?
Yeah.
Okay. See,
Dr.
I come
Shastri.
from the I come from the background of yoga meditation. This language of the neuroscience and chemistry is a little Greek and Latin for me. But let me share you what happens and why that particular slide where different parts of the cortex get activated in speaking, listening, and all that. Now, here is the simple meditation technique I have used with number of persons. I take a simple sound, for example, a sound like tum. I say no meaning associated. Generate that sound, that is, say that sound like tum. The second instruction is listen to that sound tum with your own ears and mentally watch that sound as it comes out from inside the body to outside. So there are three different engagements which happen. And wherever I have tried this particular thing, in a matter of less than 2 minutes, the person's consciousness, when this is done in waking state, in presence of several persons, okay, person standing, sitting, or just lying down comfortably, the person gets very, very deep relaxation, and in about 15 minutes, no other intervention made, several types of healings have taken place. It's recorded with a Buddhist monk, with a senior person, a medical person, 90 years, who had this palsy and all these kind of things. So they said, I will not promise anything, but let's try. So the sound was not a mantra. There was no meaning for that. All that they had to do was to repeat, to generate that sound internally, not like music, just articulate that, like give the space, and then when it ends, take it further. And in a matter of less than 2 minutes, the person's physiological relaxation state changed. Many have gone to more than 15, 20 minutes deep sleep, and wherever there was deep, I don't know what kind of pains, they have disappeared. When I have used it for the guided meditation with waking state, people have reported amazingly pain relief. Now, what I could understand from that slide is the effect could be due to the not just listening by the ears, but the whole brain and body getting activated. So this is where my interest comes. This is not mantra. This is just getting engaged with own sound. This is one of the yoga techniques. So I would like to know.
I think he mentioned that. He did mention about the temporal clustering. I think what you are saying is that this different waves that we have in the brain, so those waves can be synchronized. So the question is, how do you synchronize them? Whether by soft music, by slow repetition. So different objects may take different stimuli for synchronizing them. But naturally, the brain noise is non-synchrony. So all those, lately we have also the brain waves are coming into picture. So basically, as long as we keep the brain in control, reduce the noise in them, which is noise. Noise is flutterings. So once we reduce them by virtue of self-organization, Bela used the word self-organization. So self-organization is self-clustering. Now, self-clustering is an important attribute to the brain neural structure in the sense that once one cell silences, other cell also try to silence, and then whole cluster silences, and then it goes into different rhythm. So what you are saying is that a process that can be created where if you trigger a silencing mechanism, the whole brain can go silent, and then reducing metabolism and increasing energy and whatever else you are trying to say, healing, that kind of. But speech is different. Speech is
Speech is
definitely
far
an
advanced.
agitated
Speech is
process.
far advanced. Yes.
Agitated process in the sense you are trying to probe the brain. So we're increasing the waves to go around. Where is it? Where is it? Where is it? Where is it? And until you satisfy yourself. So basically, the next meeting that we have, we'll try to do some energy calculations, how the brain takes a signal and try to apportion it such that it can come back to its normal state. So speech becomes an energy dissipation mechanism to how do you bring the brain normal by reducing the energy. And so speech is the energy that will go out of you. So anyway, so I'll try to share, but it's a beautiful example you gave. And I think there is papers coming out about this synchrony. We don't know. There may be some methods in which what synchrony may work. I don't think there is any resonance frequency or so on has been developed. So this so-called your noise or mantra that people are talking about, how that connect to the brain, that has to be studied probably to see what frequency works, why it works. We have not done that. But we can invite other questions. Anybody else has a question? Bela, you have any comment on his observation?
So I think that the energy that the sound wave carries to the cochlear receptor, the hair cells, is the energy that the biological system can use to transfer these sound wave electrical signal and push the electrical signal along the axons and and the as the electrical signal going through the past segment of the axons coming down, so getting back to the original function, which has a different level of entropy. So as the activation is happening, it means the entropy level is different. So as as I think that's the electricity conducted in the copper wire or aluminum wire, as the origin of the electricity connected to the wire, the so-called loose electrons getting very intensely vibrating, and this vibration from the original point pushes the so-called electron frequency to the neighboring electrons get also resonate function. And then when it is going on forward, the electron, the first electron resonate stop resonating and getting the normal function. And we can see this happening when the electrical source shut off and the electrical conductivity stop, so the electron frequency is stopped, and then the wire which was warmed up is cooling down, and then the whole atomic structure is just getting back in the normal original condition. So
Dr. Mohanty, anything else from you?
No, I'm okay. I'm still learning. This is speaking and generating is big things. I see a highly compact cluster, highly delocalized cluster in the generating. That means different parts of the brain are coming into picture when you are generating this new ideas or new word. Somebody else, some other parts of the brain. I see the
right. See,
quite different.
Yeah. So see, the question is what we'll be actually in a different meeting we'll be discussing today. What is a word? Yeah. How does a word come to be? Yeah. When we say something is a word. So basically what we see, there is no word. So there's only sounds, because we can only create our phonetic sounds, which are depending upon our mouth structure and tongue position, teeth. So certain sounds we can do. So the question then is, so those sounds like the cows do, or the dogs do, or the tiger does, or cat does, we do. So then, which means that we have the ability to convert the brain signal into those acoustic sounds, which we can call our own dhvani, our own syllables. So much we can say. That is conversion. That is the word, or the tree can be a tree inside of us, and tree is scattered through the brain in different forms. The tree structure, texture, its leaves, its strength, its grounding, its population. So a tree, so all that is in many different parts of the brain. So I bring them all together. And then I see the tree inside my brain. But then the question is, I try to articulate, and I don't know what to articulate. So then the brain articulates it the way it thinks it articulates. So there is a one-to-one, some kind of a connection between the inner brain structure and the articulation method. So that we think could be the origin of the first word or first sound, acoustic sound. Then those acoustic sounds have to be parged, and we don't know who parged them and how they get, or what's the mechanism parging. So through those parging, you will say we have a word. So how do scientifically show this is our goal. But we shall see, and then the why there are so many different languages, so many different words, and how those words came to be. So that has also to be understood. So what we saw today is the brain structure where all this happens in various ways. And we'll try to see some kind of pathway through there where the brain images are converting into acoustic signals. And whether those acoustic signals are same as the signals given by the Indian text. Why Indian text? Because nobody else is giving phonetic signals. So whether that's a complete set or not, and that we are trying to experiment ourselves with a different group to see whether we have a set of acoustic signals. How complete are they? So we shall see. So anybody else has a question? Otherwise,
just to share the experiment or the practice I tried out at various places, it works its magic only under two conditions. First, the person who's practicing should generate the sound from inside and engage with that sound by himself. When practitioners try to find a shortcut, recording it and feeding through the ear using a record like this one, it did not work. So the key is you have to generate your own sound and then listen to that and engage with it. But no meaning, no meaning. That is the key thing. And the magic works. Now, how to go from sound, or what we call as the dhvanima or the phoneme, which was mentioned in one of the slides, to the next level of what we technically call as the word components, the base word, affix, process, prakriti, pratyaya, all that, memory related things. It's far more advanced. If Panini or somebody spoke of these things in the Indian tradition, how they got it, I'm not going to question that. But today, how are we going to verify or validate that understanding in today's language with today's machines? This is where I'm deeply interested with Mishraji's project.
Right. I should also comment on this thing is that when you say you create a sound which has no meaning, so that's not a scientific statement. All sounds have meanings, except we don't know the meaning.
Yeah.
The sound.
What I meant is in a very practical, simple way.
So that's what I said, the operational part and the sublime part. So the brain knows the meaning because any stimulus when it comes, the brain is mapping it completely. So it basically, and then the question is, we as persons, then we don't care about them because we don't understand it. We don't need for it. But then we can also, what this generating work does is try to explore them, the nuances, the subtleties, very small harmonies, the details about it, neural energy, and so on, and says that, can I really recall? Can I really get it? And we shall see. I think I'm glad that you are all so engaged in this. We thought 10 years ago that it's a bogus way to look at things and so on. Not many people are interested. Not people are still interested because too complex. Too complex.
May I have a comment to this?
Well, please do.
So it is very interesting that you said, and I remember three different examples. One is Mozart, the other is Beethoven, and the third is Bob Dylan. So Mozart said when he was asked, how do you compose these different symphonies? And he responded, I hear inside my brain. And Beethoven, who was deaf in the later years, and he composed the Ninth Symphony as a deaf person. So actually, he didn't hear the sound, but he heard or sensed the sounds inside his brain. So it's very interesting how the brain is working. And Bob Dylan was also asked, how did you wrote this text, your song? And he told, it just comes suddenly. I couldn't repeat it, and I couldn't write it again because the words didn't come again in the same sequence. So it's just unbelievable how our brain is working and creating this different sense. Yeah, the yoga is interesting. Oh, yes.
All right. Okay. With that, I think very positive note. That how do the ideas come? So let us end the talk again. Let us applaud by removing the mute signs, and then
Thank you.
then we'll meet in 3 months, and I'll let you know our next lecture. Thank you all. Thank you all for coming.
Thank you.
Great,
Thank you.
great afternoon in US and great night back in India.
Thank you.
Yeah,
Thank you.
namaskar.